Okay, so the groundwork for sequencing the human genome began really early on in the 20th century. After the discovery of the double helix structure by two male chauvinist pigs, by James Watson and Francis Crick in 1953, researchers started to understand that DNA was the carrier of genetic information. Throughout the 1960s and the 1970s, sige fine, thank you na rin sa kanila, um, new methods were developed for DNA manipulation and analysis. So that's when we started inventing the use of restriction enzymes. Well, not really inventing, more of hijacking the use of restriction enzymes, and also inventing the polymerase chain reaction, which allowed us to amplify DNA sequences for study. Um, in 1977, Frederick Sanger developed a method for DNA sequencing that we still call Sanger sequencing to this day. So thank you so much, Manong Fred. It's considered revolutionary but laborious. And as we've been discussing for the past couple of weeks, it can only sequence small fragments of DNA accurately. And this method dominated the field of sequencing for more than 2 decades. Again, it's still being used up to now. And it enabled the first sequencing of entire viral genomes as well as small bacterial genomes. From there, we have the Human Genome Project from 1990 to 2003. And again, we've talked about this before, but it's always good to look back, no? The most significant, and this is considered to be the most significant milestone in genome sequencing. This was considered very ambitious at its time because it was first a collaborative international research initiative, and they wanted to map the entire human genome. And what they wanted to do was to sequence all 3.2 billion base pairs of human DNA. They wanted to identify all human genes and, of course, store this information for future research. So the Human Genome Project relied on Sanger sequencing technology, and what they did was they used a shotgun method, meaning they broke down the genome into small manageable fragments. And these fragments were sequenced individually and then reassembled computationally to reconstruct the entire genome. Now, given that this project was started during the 90s, they encountered some technological challenges. For example, computing power and storage were considered to be major hurdles. It also required the development of bioinformatics to handle vast amounts of sequencing data and to align sequences properly. International collaborations, however, ensured the coordination of research across the major countries involved, which was the US, the United Kingdom, Japan, I think France, and Germany. There were others, pero sila yung major. Now, in 2003, the Human Genome Project successfully produced a near-complete, so hindi nila na 100%, just a near-complete sequence of the human genome. And the project cost roughly $3 billion. Grabe, no? And then we entered the next generation sequencing era, which occurred mid-2000s up to the present. So technological advances after the Human Genome Project marked the transition to NGS technologies. And this time it's allowing us massively parallel sequencing of millions of DNA fragments simultaneously. So to review the key platforms, we have Illumina sequencing, which was derived from Solexa technology. So this is still the dominant method up to today, and this technique uses a reversible dye terminator technology that sequences millions of fragments simultaneously with high accuracy. And then there's the soon-to-be extinct Roche 454, and this is one of the earliest NGS methods. It used pyrosequencing and enabled faster sequencing than Sanger sequencing, though it had limitations in read length and cost, and that's also one of the reasons why it's about to get extinct. Then there's the ion torrent method, which sequences DNA based on the detection of hydrogen ions released during nucleotide incorporation. And right now, it's one of the cheapest and fastest methods of doing NGS sequencing. And then we have the PacBio and Oxford Nanopore technologies, which are considered to be third generation sequencing platforms that sequence long reads of DNA in real time. And some of them are even portable. So NGS technologies significantly reduce the cost and time of sequencing. And by 2015, that's, I think, what, 9 years ago? The cost of sequencing a human genome has dropped to less than 1,000 US dollars. Diba? Imagine $3 billion before. Ngayon, $1,000 na lang, pwede ka nang mag-sequence ng human genome. So, ano-ano ba ang applications of genome sequencing? So since we'll be talking about diseases next week, let's focus on applications of genome sequencing that are non-health related. So let's first talk about forensic science. So forensics is the application of scientific principles to investigate crimes and legal matters. So this often involves the creepy analysis of physical evidence collected from crime scenes. Grabe no, sobrang uso ngayon yung pamparelax na manonood ka ng mga crime investigation shows. I've tried doing it. Na-relax naman ako habang nanonood ako, pero parang I had nightmares after. So I'm not sure if it's a healthy thing to do all the time. Anyway, so when it comes to forensic science, I guess one of the most useful advancements that came from genomics, allowed us to increase the accuracy, the speed, and reliability of identifying people. So this helps solve cold cases. And it also, pinapalaya niya or napoprove innocent niya yung mga wrongfully convicted people. So genomics has enabled the forensics community to develop sophisticated methods to analyze DNA, which has revolutionized criminal investigations. So, fun fact, part of my dissertation was actually done in PNP sa Camp Crame, even though I was working on crabs. One of our PhD students back then, classmate ko, was a forensic scientist for the PNP, and she had limited funding to do some portions of her molecular research. So, her advisor was Doc Menchi, who was my PhD advisor as well. Nag-trade off kami. We'll cover some of the costs of her PhD, pero I get to use yung microsatellite reader nila sa PNP. So I have first-hand knowledge na napakaganda po ng forensics lab natin. However, there are very, very few forensic scientists who are trained to use them. So nagkakaroon sila ng backlog ng process kasi parang 3 or 4 lang sila na marunong gumamit. And yet, nationwide yung pagsasubmit sa kanila ng tissue samples for processing. So, 24/7 gumagana yung molecular lab na yon, and they never turn off their radio kasi since forensic lab siya, human remains yung nakastore sa freezer. So, pag gabi daw, tas walang radyo, minsan kung ano-ano yung maririnig mo. So, they never really turn off the radio. Like, the entire building, laging may music na nagpe-play. Anyway, moving forward. So, ano-ano ang applications ng genomics when it comes to forensic science? So, I guess the most obvious one would be DNA profiling. This was first developed in the 1980s and remains to be one of the most, I guess, important tools in modern forensic science. So, it involves analyzing specific regions of DNA that are highly variable between individuals. And ito yung portions na tinatawag nating short tandem repeats or STRs. The unique sequences of STRs can be used to create a DNA profile, a DNA fingerprint that is nearly unique to each individual. So forensic scientists can use these profiles, these fingerprints, to compare DNA samples from crime scenes. So they collect blood, hair, skin cells, sperm cells, and so on. And they compare the DNA extracted from these samples to samples from suspects or DNA databases. Kaya one of the advocacies of the PNP is magkaroon ng DNA database ng Philippines. Pero syempre may ethical issues siya regarding privacy, identity theft, and so on. So, in the US, meron silang tinatawag na CODIS, yung Combined DNA Index System. And this was established by the FBI. It's a DNA database that allows law enforcement agencies to match crime scene DNA with profiles of known individuals or previously unsolved cases. Now, when it comes to traditional DNA analysis methods, they focus on relatively small segments of DNA. Pero dahil meron na tayong NGS, we can now analyze much larger portions of the genome, and so we end up getting more detailed information. So NGS can sequence entire genomes or even the whole mitochondrial DNA, or even single nucleotide polymorphisms or SNPs. So through NGS, we've increased the resolution and accuracy of DNA analysis, and this enables forensic scientists to extract information even from highly degraded samples or even trace amounts of DNA. So mas na-refine niya yung ability natin to collect information even though mas konti yung tissue na meron sa crime scene. Now, there's also the use of mitochondrial DNA. Again, what's interesting about mitochondrial DNA is that it's generally maternally inherited and it does not recombine like nuclear DNA. So this makes it highly useful in cases where nuclear DNA is unavailable or degraded, such as with old or skeletal remains. Since mitochondrial DNA is passed from mother to offspring, it's still useful kasi pwede natin siyang gamitin to trace maternal lineage and identify individuals in missing persons cases, as well as to investigate familial relationships. So useful siya kung gusto nating malaman kanino yung nahanap na katawan and so on. How morbid. Forensics also involves Y chromosome analysis. So analysis of the Y chromosome is used in cases where male-specific DNA is needed, such as in sexual assault investigations. So the Y chromosome obviously is passed from father to son and remains relatively unchanged across generations, making it useful for identifying male lineage or linking male suspects to crime scenes. So this method is particularly helpful when investigating mixtures of male and female DNA, such as in rape cases. I'm sorry if this is triggering. Kasi yun nga, it can specifically target the male DNA without interference from the female DNA in the sample. Okay. Now this one's pretty cool. There's also touch DNA. This involves analyzing very, very, very small amounts of DNA that's left behind from skin cells on objects that were merely touched or handled. So the sensitivity of modern DNA extraction and amplification techniques allows forensic scientists to analyze these trace samples, which are, sometimes hindi mo nga makikita na may sample. So this advancement helps solve cases where there is no visible biological material, making DNA analysis feasible in a wider range of scenarios. Although ang worrisome sa touch DNA is it can have a tendency of cross-contamination. So mahirap din mag-conclude agad-agad kasi baka dumaan lang naman yung person dun sa area. So you have to take the results of touch DNA with a grain of salt. Another application of genomics to forensics would be familial DNA searching. So sometimes a direct match to a suspect's DNA is not found in DNA databases. but familial DNA searching can be used to identify partial matches. So this method involves looking for DNA profiles that share significant similarities with those of family members already in the database. Pero paano no kung walang kamag-anak sa database? Pero yun nga, kaya important din na magkaroon tayo ng maayos na genetic database if we really do want to make sure that forensic science works. However, this also requires a huge trust in the systems that govern us, which is, in the case of our country, wala tayong masyadong tiwala. So, ang hirap din ipagkatiwala ang ating genetic information sa ating police force. Anyway, so when you're doing familial DNA searching, you look for um, you look for yun nga yung patterns na magkakamuka, assuming na merong kamag-anak yung person dun sa database. And so far, in the US at least, this has been used in some high-profile cases, including yung identification ng Golden State Killer in the US. So they used genetic genealogy to identify who that killer was. Okay? But wait, ano ba ang genetic genealogy? So this combines DNA analysis with traditional genealogical research to find relatives or ancestors of a person. So this makes use of direct-to-consumer DNA testing services. Yung gusto mong malaman kung anong ancestry mo, I forgot. 23andMe, I think. Uso yun sa US. So, hindi nila, I'm not sure if a lot of people who've used it are aware na yung database ng 23andMe ay may access ang police, ang FBI. So, you don't just get to find out who the nationality of your ancestors are. You also contribute to the genetic database that allows the police force to find criminals. So anyway, so yun, it makes use of direct-to-consumer DNA testing services, and law enforcement agencies basically use this technique to track down suspects by identifying their relatives and working backward to find the individual who left DNA at the crime scene. I mean, obviously, they don't just use the DNA, ha? Nagtitingin din sila ng mga alibis, ng mga observations, kung nakita ba yung person na yon sa crime scene, and so on. So far, this technique has become a powerful tool in solving cold cases where DNA evidence exists, but the individual's identity remains unknown. So right now, they're hoping that these kinds of services become more popular para mas dumami pa yung available information sa internet. Okay. Now, there's also a process called DNA phenotyping. It's one of the newest fields of forensics. And what they do is they use genetic information to predict physical characteristics such as eye color, hair color, skin color, and facial structure of an individual. This is very useful in cases where there are no known suspects or witnesses. And companies like, ang mga sikat dito yung Parabon Nanolabs, and they use advanced algorithms to create composite sketches of individuals based solely on their DNA. And this gives investigative leads in cases where traditional methods have failed. However, yun nga, medyo malaki ang error nito because of the balancing of both genetic and environmental influences. And then, of course, the cost of these kinds of services have a tendency to be high. So far, forensics also makes use of epigenetic analysis. So, epigenetic markers can provide information kasi on a person's age, their lifestyle, and their environmental exposures. So, in forensic science, epigenetic analysis can help estimate the age of a person from a DNA sample or even differentiate between identical twins. Kasi di ba yung twins, pareho yung genetic profile nila, but it's actually possible that they have different epigenetic modifications. So even in forensics, epigenetic analysis can help identify people or solve crimes. So just to sum up some of the applications of genomics when it comes to forensics, again, through genomics, we allow investigators to solve complex crimes kasi they can link suspects to the scene through DNA evidence, even from small or degraded samples. Pagdating naman sa cold cases, advances in DNA technology have allowed for the reopening of cold cases, leading to breakthroughs in unsolved crimes through DNA reanalysis and familial searching. And then genomics has also been used to identify remains in cases of missing persons or mass disasters, providing closure to families. And then also, genomic advancements have led to the clearing of individuals who are wrongfully convicted of crimes through the reanalysis of DNA evidence. So beyond health, genomics has certainly helped improve our lives, even in the field of crime investigation. So ano pa ba yung pwedeng application ng genomics pagdating sa tao beyond health and forensics? Well, there's also evolutionary and population genetics. So these are branches of genetics that study how our genetic variation within and between populations of organisms, so in this case, humans, changes over time due to forces like mutation, selection, migration, and genetic drift. So in humans, recent advancements in genomics have dramatically expanded our understanding of our evolutionary history, migrations, adaptations, and genetic diversity. So in a way, it helps us figure out our identity as biological organisms. And it also allows us to, I guess, envision our future. Saan ba patungo ang evolution ng species natin? Okay. So with the help of genomic evidence, we finally see the high likelihood. So the hypothesis has not been debunked and has been continuously supported, yung out of Africa hypothesis. So this hypothesis says that all modern humans, Homo sapiens, originated from a common population in Africa around 200,000 years ago. And genomic studies show that modern human populations share a common African ancestor, and then we migrated out of Africa between 60,000 to 80,000 years ago. And recent genomic analyses have allowed us to trace migration routes and timing, and this gives us a clearer picture of how early humans spread across the globe. Now, please take note, everybody, that this doesn't mean na 200,000 years ago, nasa Africa lang lahat ng tao. It's possible that there were also Homo sapiens in different parts of the world, except that the descendants of those human populations, yung mga anak nila, hindi na nag-survive. Ang mga nakapag-survive lang ng mga anak-anak ay yung human population na makikita sa Africa 200,000 years ago. Okay? And tayo yon. Kasama tayo doon. Lahat tayo ay magkakamag-anak if you trace it back to 200,000 years ago. Now, beyond that, another one of the most significant discoveries in human evolutionary genetics is the evidence of interbreeding. Sobrang, what? Ng mga tao dito. There were interbreeding between early Homo sapiens and archaic human species, such as Neanderthals and even Denisovans. So, genomic data from ancient Neanderthal and Denisovan fossils show that modern non-African human populations inherited 1 to 4% of their genomes from Neanderthals, while some populations in Asia and Oceania carry up to 5% of our DNA from Denisovans. So meron tayong konting hybrid na ibang species. So these findings reveal that rather than complete replacement, we actually interbred with archaic species. And this helped increase our genetic diversity as a species. Anyway, beyond that, genomic studies have also uncovered signatures of natural selection in our genome. So this reveals how we've adapted to different environmental challenges. So yung iba dito na-discuss natin sa, kung naabutan nyo yung worksheet sa ecology on evolution, on human evolution. So, medyo tinanggal na kasi siya in more recent versions. Then you would be familiar with some of this. But anyway, first one, lactase persistence. So, in populations that domesticated cattle and relied on dairy products, selection favored the continued production of lactase. So, this is the enzyme that our digestive systems produce to break down lactose into adulthood. Now, in most other populations, you lose your ability to digest milk as you grow older. That's why in Asia, for example, marami sa atin ang lactose intolerant. So anyway, this adaptation is most prevalent in populations from Europe, the Middle East, and parts of Africa. So it's actually more common to be lactose intolerant as an adult, except if you're from Europe, the Middle East, and parts of Africa. Okay? Next, when it comes to altitude adaptation, populations living at high altitudes, such as Tibetans and Andean peoples, have evolved genetic adaptations to low oxygen levels. In Tibetans, this adaptation involves variants in the EPAS1 or the EPAS1 gene, which helps regulate the body's response to hypoxia. And then even in malaria, no, meron na rin tayong nakikitang evolution related to it. So in regions with a high prevalence of malaria, mutations in genes like HBB, which is actually linked to sickle cell disease, and G6PD, and meron ding disease na tinatawag na G6PD deficiency. These mutations in these genes provide some protection against malaria, demonstrating how infectious diseases have driven human evolution. But in the Philippines, meron din tayong unique population that's showing traces of human adaptation, yung Badjao population, who are showing physical adaptations that allow them to stay underwater longer. So they're capable of holding their breath for much longer. They're capable of using their eyes efficiently underwater. And there are signs that this is linked to the selection of genes that contain certain mutations that allow them to have increased lung capacity and adaptations in the production of yung nagmo-moisturize sa mata natin that allows them to see underwater. So yeah, those are some of the things that people in evolutionary genetics are studying when it comes to the human species. Beyond that, we've also seen that human populations show varying degrees of genetic diversity, and this is shaped by our evolutionary histories. For example, African populations exhibit the highest levels of genetic diversity kasi sila yung source. And yun nga, the fact that they're a source shows that they have a lot of unique mutations that make them more distinct from the other human populations that exist as of the moment. Of course, despite the fact that there are populations that have higher diversity, genomic studies have shown that genetic differences between human populations are still relatively small. And this supports the idea that race is a social construct rather than a biologically significant division. So most genetic variation actually occurs within populations rather than between them. So hindi talaga subspecies ang race. It's really more of a social construct. And if you look at us as an entire species, there is very little genetic difference across all of us. Now, the use of genomics has also allowed us to map human population structure more accurately. Large-scale studies using genome-wide data have revealed that patterns of genetic similarity and differentiation among populations exist. So right now, there are studies like the Human Genome Diversity Project and the 1,000 Genomes Project. Yung mga project na to have documented the genetic diversity of various human populations. So this has allowed us to get to know a little bit more yung past migration patterns, population bottlenecks. For example, one of the more established bottlenecks sa Southeast Asia is the explosion of a volcano in Indonesia. I think it's the Tongo explosion. And it eradicated majority of the human population in Southeast Asia, leaving us, the rest of the Malay populations, descending only from a single population that was living near the south of Indonesia, as far as I know. So, yun. And we also get to understand a little bit more about current gene flow. So, recent work has refined our understanding, for example, of how human populations expanded, how we've contracted over time, and how we've mixed continuously over time. And right now, some of the major migrations that are of interest to researchers would be the Neolithic agricultural revolution and also the Indo-European expansion. Another interesting paper that linked human migration to the genetics of plants would be a paper by Dr. Michael Puruganan. He's a Filipino scientist who's the dean of NYU. He has a genomics lab there. And by following the evolution or the genetic variation of rice across Asia, they were able to link it with findings from anthropological scientists to figure out the migration patterns of people in Asia. So sobrang interesting niya kasi, 'di ba? Rice is life for us, and even when you cannot get access to human DNA, you can see the human impact on the genetics of the organisms we consume, the organisms we grow. So ang laki-laki talaga ng genetic footprint natin. Kaya napaka-interesting ng mga ganyang paper. Anyway, speaking of gene flow, modern genomic techniques have also traced the gene flow between populations across continents. So for instance, studies show that there was significant gene flow between African and Eurasian populations throughout history, particularly during key periods of human migration. So the genomic signature of ancient humans, such as hunter-gatherers, early farmers, and steppe pastoralists. So ang steppe pastoralists, sila yung nagpapastol ng mga hayop sa mga medyo highland areas. So these were detected in the genomes of modern populations in Europe and Asia. So this mixing of genes reflect the complex demographic history of human populations. Sadly, because I guess the white population have greater access to these technologies, syempre mas maraming information regarding their ancestors. And the dream is that, more studies can be done in Southeast Asia, so that we can figure out din sa atin, ano nga ba yung historical and current migration patterns of the people who we are more genetically linked to. Okay. And then, so when it comes to evolution, while many aspects of human evolution occurred in deep prehistory, obviously, genomics has shown that human evolution is ongoing. For example, the ability to tolerate high starch diets has evolved in agricultural societies with selection for multiple copies of the amylase or the AMY1 gene. Actually, related siya dun sa gluten allergy or gluten intolerance. So in countries, for example, where they don't have a lot of starch, nagkakaroon ng increase in number of people who have gluten allergies, so making them gluten intolerant. And yet for societies where starch is very common, kung saan maraming access to gluten, we've evolved to not have that mutation. And so we can enjoy gluten as much as we want without any bad effects. So yay! Sorry. Um, but apart from that, genomics has also shown evidence of recent selection for traits like skin pigmentation. So, in countries where a paler skin color is preferred, mas nagiging lighter sila over time. Whereas in countries where a more tan color is preferred, mas nagiging colored sila over time. And of course, what's interesting here is our skin pigmentation actually protects us from skin cancer. And so selection towards a lighter skin tone is not necessarily a good thing. No? So I love being, alam niyo yung kwento ng matanda dati. Well, I don't know if you've heard about this, pero there was this ancient Filipino story na nung ginagawa daw ni Bathala ang mga tao sa mundo, kumuha siya ng clay, tas binake niya. Kaso yung first batch daw, na-distract kasi siya. So nasunog. And out came the dark-skinned people of the African areas, African regions. Yung susunod daw na batch ng tao na binake niya, dahil ayaw na niyang masunog, agad-agad niyang hinango. So it ended up being pale, and hindi rin siya naging happy. So yun daw yung mga tao sa western areas. Yung third time na ginawa ni Bathala yung tao, alam na niya kung ano yung dapat gawin, and out came the beautiful brown-skinned people of Southeast Asia. And interestingly, when it comes to the genomics of skin cancer, our skin tone is perfect in terms of protecting from skin cancer. So, I love our color, and I'm very, very proud of our skin color. So, no need to breed us into paleness. Anyway, apart from skin pigmentation, we are seeing selection for immunity to certain diseases and even brain development genes. So this is in response to new environmental pressures and cultural shifts. And then we've also detected or started detecting genetic drift and bottlenecks. Sorry, I had to cough. Anyway, so genetic drifts, just to remind everybody, refers to random fluctuations in gene frequencies that can occur in small populations. So population bottlenecks, such as those caused by environmental disasters or migrations, which are still happening up to today, reduce genetic diversity and amplify the effects of drift. So, isa sa mga pinaka-alam na example, I think nabanggit ko na ito kanina, is yung Toba volcanic eruption. So, this is called the Toba catastrophe theory, which suggests that a volcanic eruption about 74,000 years ago caused a severe bottleneck in the human population. So genomic data supports the idea that bottlenecks occurred in human history, but of course, the extent of their impact is still debated. Kasi our genetic data will tell us when it most likely happened, but we still need the help of archaeologists, anthropologists, geologists, to see if there is environmental evidence of ano yung nag-cause ng bottleneck. Kasi for all we know, hinuli lang pala tayo ng mga aliens. Hinarvest nila tayo kaya nagkaroon ng bottleneck. So without the help of the physical sciences, haka-haka lang or hypotheses lang or myths lang yung nakikita natin sa genetic data. It really needs confirmation or validation from our physical scientists. Okay. Now, of course, when we're studying the human genome, it has a lot of ethical implications. So when it comes to privacy and genetic data ownership, um, I've been mentioning it earlier nung forensics discussion natin, but the large-scale collection of genetic data raises concerns over our privacy. We have ongoing debates over who owns genetic information, whether tayo ba, yung individual, is it the medical institution who paid for it, or is it the private companies, again, are capable of paying for it? And misuse of this data, such as in insurance or employment, could eventually lead to genetic discrimination. And then, informed consent. For genome sequencing, ensuring proper informed consent is complex. Patients must fully understand the scope of what sequencing can reveal, including incidental findings such as genetic predispositions for unrelated diseases, parentage, like, oh my God, hindi pala yan yung tatay mo. And yun nga, yung risks of data sharing. So lahat yan, kailangan ma-include sa informed consent form. And then, there's data security and sharing. The huge amount of genetic data that's being generated would require very, very strong cybersecurity to prevent breaches. And while data sharing is essential for advancing research, it also poses a threat to individual privacy if we don't handle it properly. And then, of course, for countries like ours, na ang laki ng divide between the rich and the poor, we have to ask about equity and access. So the benefits of genome sequencing are still not uniformly accessible. Countries like ours often lack the resources to benefit from genomic medicine. And so this makes global health inequalities even bigger. Parang lumalaki pa lalo yung divide. And the cost, although nagmumura na siya, can still be prohibitive for many people in low-income settings. Tapos, pumasok pa yung gene editing, tsaka CRISPR. We can't really separate genome sequencing kasi and genetic engineering. So, CRISPR-Cas9 technology allows scientists to edit DNA with precision. And so, ethical concerns have arisen over the use of gene editing, particularly in germline cells, where changes can be inherited by future generations. This debate became more prominent after He Jiankui, yung Chinese scientist, claimed to have edited the genomes of twin babies to be resistant to HIV. So this raises questions about designer babies, eugenics, and all of that. And then, of course, all of these possibilities would have a psychological impact on us. If you discover your genetic risks, if you find out you're susceptible to like incurable diseases such as Huntington's, ang laki nung magiging psychological impact nun. So managing the dissemination of sensitive information like this requires careful counseling and ethical considerations. Okay? So now let's move forward to understanding structural variations found in our genome. So what do I mean by structural variations? These are large-scale changes in our genome that involve duplications, deletions, inversions, or translocations of segments of DNA. So these variations contribute to our genetic diversity, but they can also cause disease susceptibility, but they can also help with our adaptation. So ano-ano ba yung mga common structural variations na meron tayo? So first, we have copy number variations. So these refer to segments of DNA that are duplicated or deleted, leading to differences in the number of copies of certain genes or regions between individuals. So copy number variations are common in the human genome, and they can have significant phenotypic effects. So hindi to silent, no? Pwede silang magkaroon ng physical effect sa atin. So kanina nabanggit ko yung starch. So our example would be your amylase gene, AMY1, AMY1. So this helps digest starch. And we see copy number variation between populations. So populations with high starch diets, such as agricultural societies, have more copies of the AMY1 gene, allowing more efficient starch digestion. So copy number variations in certain regions have also been associated with diseases such as schizophrenia, autism, and autoimmune disorders. Okay. Next, we have inversions. So this occurs when a segment of DNA is flipped in orientation within the genome. So this can disrupt gene function or alter the regulation of nearby genes. So some inversions may be beneficial and have been linked to adaptations. So for instance, an inversion on chromosome 17 has been found to be more frequent in Europeans and has been associated with increased fertility in certain populations. I guess beneficial siya for them kasi nag-shrink yung population size nila. Sana sa atin baliktad kasi ang bilis lumaki ng population natin eh. Actually heard one speaker mention na the Filipino population is one of the youngest populations in the world, which means mas maraming bata na pinapanganak kaysa mga taong namamatay. This is often looked at as a good thing by majority of the more developed countries because their population size is shrinking and they don't have enough workers to support the social services they give to old people. But in our case, since we have limited resources, I don't think the Filipino population wants to continue expanding its population. Or siguro ano talaga, hindi ko alam, guys. Maybe we need a reversal of this inversion so that we don't end up being too fertile because it's harming our country. Anyway, there's also indels. Hindi incel, guys, ha? Indel, hindi incel. Indels, insertions and deletions. This refer to the gain or loss of DNA segments in the genome. So these structural variations can range in size from a single base pair to large stretches of DNA, and they can significantly affect gene function. Again, hindi rin po ito silent. So for example, we have deletions in the CCR5 gene. Again, CCR5 gene. And this confers resistance to HIV infection, as the virus uses the CCR5 receptor to enter cells. So individuals with these deletion are less susceptible to HIV. Certain deletions naman have been associated with conditions like Williams syndrome, which is a developmental disorder. So pwede siyang maging okay, pwedeng hindi. It's a mixed bag. And then there are translocations. So this occur when segments of the DNA are swapped between non-homologous chromosomes. So this can disrupt gene function and lead to diseases such as cancer, where translocations activate oncogenes or deactivate tumor suppressor genes. Okay. And then, of course, there's SNPs, the ones that we use most frequently. These are the most common type of genetic variation and occur when a single nucleotide is substituted for another. So while SNPs are considered small-scale variations compared to most of the other structural variants we've discussed, they play a significant role in human evolution and diversity. Kasi SNPs can influence traits like blood type, disease susceptibility, and drug metabolism, just like what we discussed sa GWAS. Kasi 'di ba yung GWAS, through GWAS, we have identified thousands of SNPs linked to common diseases such as diabetes, heart disease, and cancer. Now, one other example that I don't think we've discussed in the lab is that SNPs in the EDAR, the EDAR gene, are associated with thick hair and increased sweat gland density in East Asian populations. So this illustrates how single nucleotide changes can lead to evolutionary adaptations. Okay, so I think that concludes our lecture for this morning. It's relatively short. Babawian ko kayo sa human diseases kasi maraming sakit. But just to help prepare you for this coming Thursday, the output for this particular module is group presentation once again. So you need to prepare a 10-minute presentation, this time on the latest advancements in human genomics that are not directly related to disease. Your presentation should highlight recent developments and innovations in the field, demonstrating their significance and potential impact on our understanding of human genetics. So please use peer-reviewed journal articles that were published within the last 5 years and focus on advancements in human genomics that do not pertain to disease, but rather to other aspects such as evolution, adaptation, or non-medical applications. Now, if you look at the People tab in Animospace, this would be under reporting number 2. So since we have time, let me go through who your group mates will be. So for group 1, you are Cachero, Campos, Chan Sofia, Nang, Ramirez, and Reyes. For group 2, you are Akyatan, Bata, Luna, Po, Sison, and Yao. For group 3, you are Kadoy, Cao, Olis, Pateño, Velasco, and Villaflores. For group 4, you are Acuña, Gonzalez, Pambid, Portento, Rebanal, and Urbi. Group 5, you are Bao, Denya, Flores, Gako, Lim, and Ke. And for group 6, you are Aban, Madrid, Nullias, Ong, Sison, and Tan. So again, you should be able to see this also in Animospace. When you click People tab, just look at reporting number 2. So the recommended presentation structure would be the following. So don't make it too complex, but you should provide an introduction. So this gives a brief overview of human genomics and its relevance to a non-medical field. And then give us an overview of the advancement that's covered in the journal article that you've identified. So describe the advancement or the discovery. Make sure to include details about the technology or method, so the methodology that was used, and also the main findings. And then highlight the significance. Why would this advancement be considered important? Discuss its implications in the field of genomics and how it contributes to our broader understanding of human genetics. Give examples, so relevant examples or case studies from the journal articles about how we see these advancements in the human population or how they can be used in the human population. And then conclude by summarizing the key points and highlight future directions or future applications of the advancement. So you will need to prepare a PowerPoint or similar visual aid to support your presentation. Just like before, make sure your slides are clear, concise, and also visually engaging. And make sure that you practice your presentation to stay within the 10-minute time limit. Now, we will be assigning you your group topic. So no need to start doing your research now because during your face-to-face meeting on Thursday, you will be assigned a particular topic where you should focus it on. And I'm hoping that you can use the rest of the face-to-face period to discuss with your group mates on how to proceed with this. And I prefer it when you use the time period in class to meet with your group mates. So there are no excuses regarding, ay, hindi ako pwede, wala ako dito. And so that we minimize the excuses made by people who don't really contribute to group projects such as this. And as much as possible, I would really want to use opportunities like this in my class to train you how to work in groups, because in the future, if you want to be a researcher or a medical practitioner, your social skills with others um, would really need to be improved in order for you to be considered a professional, in order for you to be able to create high impact innovations to help the world. So, sana po hindi tayo maging pabuhat. Now, the presentation date for this is slated to be on October 14th. So anyway, additional details will be given this coming Thursday during the face-to-face meeting for the week. Okay, and if anything else happens, if anything else needs to be announced, you can also watch out for announcements in Anima Space. Okay, so more or less that's it for today. But of course, before I close this session, any questions or points of clarification? Oh, there's a very interesting question in the chat. What is the worst that can happen upon data breaches in genetic databases? Well, right now, since we still have limited technologies, I think I think it's still related to identity theft. If, for example, people get access to your genetic information and somebody's out to get you, of course, you'll have to have really, really bad enemies here who who are really out to get you. They can plant like your genetic evidence in like crime scenes or in other situations. But I guess more realistically, in the event that you do not have enemies that are out to get you, for example, if you're trying to get medical insurance, one of the first things that they need to do is to get you checked up biologically, to see if you already have existing diseases such as diabetes or heart disease. And this will make your monthly payments more expensive. But can you imagine if they have access to your genetic data? Even before magkaroon ka ng symptoms ng diabetes or magkaroon ka ng symptoms ng heart disease, pwede na nilang taasan yung premiums mo. And ang justification nila is, ay kasi ma'am may predisposition po kayo so dapat ganito kalaki yung bayad nyo. So if you're on the side of the insurance company, parang mas malaki yung kikitain mo, you think that's okay. Pero on the side of the people who might be struggling to make ends meet, parang bakit ganito pa lang kaaga, pwede namang inaalagaan ko yung health ko, hindi naman siya guarantee na magkakasakit ako. Tataasan mo na siya agad. So pwedeng ganun. And in the even more distant future where they can start predicting our intelligence, our ability to stay fit, for example, the companies who plan to hire you, kung maka-access sila, for example, sa mga genetic databases, can use that information to say, ay, feeling ko, hindi totoong mataas yung grade mo. Feeling ko nag-cheat ka nung college kasi sabi ng DNA mo, bobo ka eh. Pwede yun. Pwedeng mangyari yun. So, those are some of the things that people are discussing in terms of the ethics of forming genetic databases. Kung baga, we are only limited by our imagination and existing technologies on what people can do with our DNA. Imagine yung nga SIM card pa lang pwede nang mang-scam yung mga tao. Paano pa kung may access sila sa DNA nyo? No, so I hope that was able to answer your question. It's a very, very interesting question. Interesting and scary. I agree. I agree. It's gonna be a strange new world in a decade or so. Gonna be very different from how it was in the past. Still exciting, but also, can you imagine yung mga nasa movies lang, yung uminom ka lang ng tubig or wine, then somebody gets your cup and extracts your DNA there. Parang, right now, parang feeling natin wala pa siyang masyadong pwedeng gawin. Pero in the future, you also have to be careful of your biological footprint. Parang, creepy. Anyway, wag naman tayo maging praning kasi tayo yung mga scientists na magpupush forward ng genomics technologies, and kung masyado naman tayong praning, wala nang technology na mag-move forward. So, just has to be a balance of being aware of the ethical issues and also still being excited about what the positive things the technology can bring. Okay, any other questions? I hope hindi masyadong malakas yung ulan sa area nyo. Dito sa may Pasay area, medyo humina-hina na siya. But just in case you're in campus, I hope it doesn't rain too hard so it'll be easier for you to go back home later. So anyway, ingat-ingat po everybody. And I hope that everybody brings their A game in the face-to-face class on Thursday. And I hope that everybody contributes to their group activities. So thank you so much, everybody. Stay safe. Bye-bye.